Welcome to chapter 12.3's video. We are going to talk about the cycle of viral replication and how we culture viruses in this lecture. So this is actually going to be a pretty short one. It's going to go pretty quick, but there's a lot of really important information in this lecture that's going to be on your next exam. So let's talk about the learning objectives for this section, and you need to be able to explain how viral genomes enter cells, so the process of the viral replication cycle. You need to be able to describe lytic and lysogenic cycles and know the difference between the two. We're going to talk about the lytic and lysogenic cycles of bacteriophages because they're a little bit easier to understand than other viruses. And then you're going to be able to explain how viruses are cultured using host cells. Okay, so one of the first things we need to talk about in this section is the process of how a virus gets into a host cell and replicates and then leaves. Okay, so this is what we call a viral replication cycle. So all viruses go through similar cycles to this. So this is kind of like the basics of that cycle in a nutshell. You need to know this cycle. You need to know the steps and what happens at each of these steps. This will be on the next exam. Okay, so the first step in the viral replication cycle is that the virus actually has to recognize a host. Okay, that cell, it looks good. It looks like it's going to be my new home. Okay, so the virus is going to recognize the host cell that it wants to invade, and then it's going to go through the process of attaching to that host cell. So a lot of this is going to go through those spike proteins on the outside of the virus. They're going to attach to receptors that are similar in shape to that spike protein, so they're going to be able to adhere and attach themselves to the cell that way. So the second step is the actual entry of the virus into the host cell. Okay, so when they attach to those receptors, it's going to send a signal to the cell that there's something that needs to be brought into the cell. So it's going to engulf it and encapsulate it, usually in a vesicle, and bring it across. Now, different viruses are going to do this in different ways. Okay, they're going to somehow going to need to be able to get out of the vesicle if they came in via a vesicle to get their genetic material into the cytoplasm. Okay, so they're going to escape the way they got in, and they're going to release their genetic material in the cell, and they're going to hijack your cellular machinery in order to go through that replication process like we've talked about. Okay, so step 3 is we are replicating the genome, we are transcribing it, we're translating it, and if it's a DNA virus, if it's an RNA virus, we're making proteins from the RNA. Okay, so what's going to happen is we're going to get all these little parts that make up the virus. Okay, so when this happens, part of this process of replicating the genome is also going to include the instructions on how to put these different parts, these different proteins together to make new viral particles, new virions. Okay, so step 3 is the assembly of the new viral particles, these progeny virions. Okay, so it's going to start to take the replicated genome, and it's going to start to package it together and put it in these capsids and these proteins. So it's going to start to assemble them. So once they are assembled, then the last step can happen, and this is going to be how the virus exits the cell. Okay, so it can go through a budding process where it goes and pushes against the cell membrane and gets released that way. It can go out in a vesicle, and then just get poured out into the extracellular space like we've seen happen to waste in exocytosis. Or we can do what some viruses do, and we can kill the host and destroy it and open it up via lysis and release it that way. Okay, so know these 4 steps. Know the 2 ways that the virions can exit the host cell. We're going to talk more about this last part here in just a second, but it's really important that you understand what is happening at these stages. Okay, these will be tested on. Okay, so let's talk about the bacteriophage replication cycle. Very specifically, there are 2 parts of how bacteriophages replicate. Okay, there's 2 kinds of cycles. We have the main one, which is what we call the lytic cycle, or a lytic infection. Okay, so this is when the bacteriophage is virulent. So this is when it's causing damage. Okay, this is where it's working on replicating itself and then spreading to as many new hosts as it can. Okay, so it's going to be a virulent bacteriophage. Okay, and what it's going to do is it's going to kill its host. It's going to cause the host cell to lyse or rupture in order to release these new virions that have been assembled. Okay, when the cell is lysed, this is what is also referred to as a burst. Okay, so a burst size is simply the number of virus particles that are released when that cell is lysed. Okay, the larger the burst size, the more viral particles are released. Okay, so this is the kind of the virulent cycle. So we're making viruses, we're replicating them, they're going on to infect new hosts. Now, the other half of the bacteriophage's replication cycle is the fact that it can go into this lysogenic or lysogeny cycle. Okay, so this is when the phage genome is going to integrate with the host cell. And when it does this, it's going to go into what is called a prophage state. Okay, so when it's a prophage, this is going to be when the prophage has the capacity to make new phage particles, but it's just not happening. It's just sitting there chilling and enjoying itself and not replicating. Okay, so when it does this, it can also bring new genes to the genome of the host, so the bacteria. We talked about this. This is transduction, right? Okay, so it's going to kind of chill until there's an environmental stress that's going to cause the phage DNA to cut itself out of the host genome and replicate itself again. So this is one of the main ways that the Shiga toxin replicates itself. We've talked about it a little bit in class. So let's look at what this actually looks like. Okay, so this is a really good slide to know. This is going to tell you the difference between the lytic and the lysogenic cycles. Okay, so our main virulence pathway for bacteriophages is we're going to have the host. The phage particle is going to come in and it's going to attach and it's going to puncture the cell, the bacterial cell, and inject its genome here. Okay, so the genome is going to be injected in its linear form. Okay, and it's going to cyclize and become that circular chromosome we know. So it's going to be like this. What's going to happen is the cell is going to use this and it's going to replicate and it's going to start to make all these different proteins. It's going to make the proteins for the capsid. It's going to start to package the DNA into this capsid. Okay, so we're going to start to make our new little viral particles. Okay, and then at some point we're going to have newly formed virions here that are fully functional, and what's going to happen is the virus is going to cause the cell to go into programmed cell death into apoptosis, and it's going to lyse and release all these newly made virions into the environment, and they're going to be available to go and infect new cells. Okay, so this is lysis. Okay, so this is when we continue to make new viral particles and we burst the cell open in order to release them. So lysogeny is going to happen when we go through these first stages. So we get to this one where we have our cyclized DNA. Okay, what's going to happen is this phage DNA is going to come and insert itself into the host genome. Okay, this is when we form the prophage. Okay, so now that the viral genome is integrated, that host cell is going to continue to divide. And every time it divides, we're going to copy that phage DNA along with it. Okay, so we're going to get more and more cells with this phage DNA. Now, something can come along in the environment. Usually it's something that's going to cause stress. So the stress is going to cause this viral genome to excise or cut itself out of the host genome. Okay, and once it does this, it's going to start to be replicated and we're going to start to read it. We're going to start to make those capsid proteins. So we're going to enter back into the lytic cycle and make new virions and burst the cells to get them released. Okay, so this is the difference between lysogeny and lytic. So lysogeny is pretty much when it's going to be dormant. It's not going to be actively causing an infection, but it's still there. Okay, a good example of what we have of a virus that does this is that herpes simplex virus 1, the one that causes cold sores. Because a lot of times that virus, HSV1, is going to integrate into the genome and go into the lysogenic cycle and be dormant. Okay, so different stressors, so it can be environmental stress, um, actually it could be mental stress as well and what we have seen in a few examples. Okay, so some sort of stress is going to act on the host, and it's going to cause the phage to excise its DNA from the genome and cause it to go back into the lytic cycle. We don't really know and understand a lot about these outside stressors, but we just know that when it happens with HSV1, it's going to cause that cold sore, and that's when it goes into its active phase. Okay, so the last thing we're going to talk about are how we culture viruses. So we talked about this with bacteriophages. We can make what is called a plaque assay. Okay, so we're going to make a plate with bacteriophages added to it. Okay, so what we're going to do is we're going to have our E. coli. It's going to be in a complex media. It's going to be in a rich culture broth. Okay, so we're then going to take the bacteriophages and mix them together. Okay, so we're going to have the bacteriophages and the E. coli in this culture. And then what we're going to do is we're going to take our agar, so what we put on our plate, and we're going to combine our tube with the phage E. coli mixture, and we're going to pour it into there, and we're going to pour it on the top of an already poured agar plate. Okay, so when this happens, it's going to spread out evenly, and the agar is going to set up and become solid. What's going to happen is we're going to incubate this overnight, and what we're going to see is we're going to see a bacterial lawn form in this top layer. Now, whenever there is bacteriophages, we're going to see them start to lyse or kill those bacteria cells. And what we see on the plate on this assay are these clear areas where there's no lawn anymore. Okay, this is the result of the virus lysing the cells. So we call these clear areas plaques. Okay, so this is one way we can culture viruses and study them. Now, this works very well for bacteriophages because this is the way that we grow bacteria. But what about viruses that infect humans? Well, the viruses that infect humans, it's going to be a little bit harder to culture them because, unfortunately, for human viruses, in order to study them actively working and dividing, is we have to have a host cell in order for this to happen. Now, I don't know if you know much about human cell lines, probably not a lot. Um, Spoiler alert, human cells don't like to grow and divide outside of the human body. So we can take our cells and we can put them in culture, and they're going to replicate about 20 times until they stop. Okay, because one of the things that our cells have during replication, and you probably remember this from biology, is the fact that we have these checkpoints at different stages of the cell cycle to stop and evaluate where our DNA is and how healthy our cell is. Now, if there's any DNA damage, what's going to happen? These checkpoints are going to stop cell division and are not going to allow cells to grow past that point. So this is what happens when you take your cells out of your body and put them into culture. So they're going to replicate about 20 times, and then these checkpoints are going to happen. They're like, there might be damage to this DNA. You're copying weird. We're just going to shut this down, and the culture is not going to grow anymore. Okay. Now, one of the times that this is not a factor is if there's a mutation in that checkpoint. So a lot of cancers have a mutation in P53, which is a molecule that interacts at these different checkpoints in the human body. And the thing that shuts this down if there's too much damage to the DNA. So with cancer cells, they're already mutated. So they've mutated this P53 protein, which means that the cancer cells no longer stop at these checkpoints. This is why it's called uncontrolled cell growth, is because it just keeps growing and dividing, even though the even though the DNA is damaged. No way about it, the DNA is damaged. Okay, so cancer cells because they have this mutation that allows them to go through these checkpoints, they're what we call immortalized cells. They can replicate indefinitely because they don't have these checkpoints, or they're not stopped at these checkpoints. Okay, so what we do when we're studying viruses is we're going to use an immortalized cancer cell line in order to do this. So what we're going to do is we're going to make a plate very similar to the one that we made on the previous slide. Okay, so we're going to have a layer of cells. Okay, we're going to put that virus into a liquid medium, and we're going to let it sit there, and we're going to let that virus infect the cells. Okay, then we're going to pour off the liquid media that we've used to infect those cells, and then we're going to add a gelatin medium on top of it. This is going to prevent the viruses from escaping. Okay, so what we're going to see is the viruses are going to reproduce, and just like with the bacteriophages, they're going to cause the cells to lyse. Okay, so we're going to start to see plaques when this is happening, when the viruses are starting to cause the host cells to die. Okay, and this is going to be what is happening in this plaque. So these areas of dark purple are the human cells. These are this immortalized cell line. Okay, so these are the healthy ones. So these light areas are where lysis from the virus is starting to happen. Okay, so another reason we put the gelatin medium on top of the cell layer is to stop the viruses from moving on to different cell. So we can see it rupturing single cells. Okay, that's what's going to give us our plaques. So these lighter areas in this micrograph are those plaques that are forming in the cell line. Now, we have to ask ourselves where we got these cell lines, and the answer is we got them from cancer patients. One of our most famous immortalized cell lines, we call them the HeLa cells, actually have a pretty sordid past. So they are called HeLa cells, so H-E-L-A, because they are isolated from a African American woman in 1951 by the name of Henrietta Lacks. Now, one of the things we need to know about HeLa cells is they're used all the time in research. In fact, in 2023, it was found that the HeLa cell lines appeared in more than 110,000 papers since they were used to make cell lines in the 1950s. That's a lot of research that uses these cells. The problem is there's an ethical issue with how these cells were collected. So Henrietta Lacks went to Johns Hopkins University to be treated for cervical cancer. They took a biopsy, so they took part of her tumor, and then they started to grow it in the lab without getting her permission. Okay, and Henrietta then died a year later, and they were still using her cells. In fact, when it came out, her family was pretty upset because Henrietta Lacks' family were living in a pretty poor part of Baltimore at the time. Okay, in fact, in 2013, so Fisher Scientific are I think it's Fisher Scientific, one of the Fisher umbrella companies, are the ones that were commercially selling these HeLa cells to different research groups. So they made billions and billions and billions of dollars on these cells that they isolated from this woman in the 1950s. Now, as of 2013, because there was such an ethical issue with how these cells were gathered and there was no consent given for them to use them for research. In 2013, the National Institute of Health here in the US came to an agreement with Henrietta Lacks' family about how to use these cell lines. Specifically, they are the ones that decide whether you get to look at the genome of the HeLa cells or not. Okay, so this is something that's protected. So that was the first step that was ever made to kind of show recompense for how these cell lines were collected. And it was buried in 2023, so we've been using HeLa cells for over 70 years now. So it was in 2023 when the scientific company that was selling HeLa cells finally came to an agreement with Henrietta Lacks's family in order to compensate them for the trauma and the amount of money that they rightfully deserved, as the fact that they used one of their family members' cells without permission. Now we don't know how much they settled for, but they did finally make things a little bit right, even though it was 70 years later. Now, if you ever want to know more about how the HeLa cell line came about, there is a very, very, very good book called The Immortal Life of Henrietta Lacks by Rebecca Skloot. Very, I'd highly recommend reading it if you would like to know about this story. Since then, there have been a lot of procedures and policies put into place to be more ethical when we can collect samples and start cell lines from samples from actual patients. So this is how we culture viruses in the lab to study them. It's not easy. It's really hard actually, but we've had a lot of medical breakthroughs thanks to Henrietta Lacks's cells, this HeLa cell line. Our lives wouldn't be the same without it. Personally, I'm very happy that we finally have kind of made peace with her family and kind of done right by them, even though it took a long time to do so. Okay, so that is 12.3. So in 12.4, 12.5, and 12.6, we're going to talk about 3 different viruses individually in each section and talk about some of their common characteristics and the type of viruses they are. So we'll see you in 12.4.